So, welcome everyone to the March edition of the IICD seminar. It's my great pleasure to introduce our speaker today is Maria Brbic. She's an assistant professor of computer science and life sciences at EPFL. So before she starts, I'm going to just give a brief overview of her background. So her research bridges AI and biology, focusing on developing new AI methods that drive advances in biology and biomedicine. Maria received her PhD from the University of Zagreb in 2019. During her PhD, she also worked at Stanford University as a Fulbright scholar and at the University of Tokyo. Prior to joining EPFL in 2022, she was a postdoc at Stanford as well. Among other recognitions and awards, she was named a Rising Star in EECS by MIT in 2021. She received an Early Career Award from SIB, Swiss Institute of Bioinformatics, in 2023, and she was awarded the SNSF Starting Grant in 2024. So with that, Maria, it's great to have you here, and we look forward to listening about multimodal generative models of cellular complexity.
Thank you so much, Simon, for inviting me, and thank you so much for everyone. Really excited to be here. I'm sorry I could not make it in person. It would be much better, of course, but hope we will still have a fun hour. So I will tell you a bit about our recent work on multimodal generative modeling of cellular complexity. So we are currently in the midst of genomics revolution that is transforming our understanding of biology at a cellular level. With advances of single-cell RNA sequencing technologies, we can nowadays measure gene expressions in hundreds of thousands of individual cells. And not only that we can measure transcriptome of a cell, we can also measure proteins, epigenome, phenome, and with multi-omics technologies, we can do multimodal measurements in the same time, at the same time in the same cells. And very latest spatial technologies enable us to look where individual cells are located within their native tissue context, while simultaneously being able to capture their gene expression profiles. At the same time, we are also witnessing an AI revolution. With the rise of generative AI paradigm, we've recently seen completely new capabilities of AI systems arising in bringing us to the era of foundation models. Today, we think of a foundation model as a model that has learned features that are transferable to a wide range of downstream tasks. Most famous examples would be large language models, such as ChatGPT, for example, that are pre-trained on internet-scale data to predict next token in a sentence, and then can be applied to a variety of downstream tasks. But a fundamental question is, how can AI help us to advance our understanding of biology? How can it bring us closer to understanding ourselves, how they function, and what goes wrong on a cellular level in disease state? And ideally, our goal is not just to make predictions, but really to use AI to discover new biological mechanisms. But unlike a natural language processing or computer vision, in biology, we often have to deal with data that is very extremely heterogeneous. And data sets, for example, are collected across different experiments, meaning they come from different tissues, different donors, different time points, different species, and also different modalities. And the ultimate goal would be to develop an integrative, holistic understanding of biology that would capture the complexity of life across different molecular scales. And here, AI technologies really hold the potential to unify these fragmented data sets and extract meaningful patterns that would be generalizable across different biological contexts. And in our prior work, we tackled some of these excesses of complexity. For example, how can we integrate data sets across different tissues, where we developed the Mars method? How can we transfer knowledge across data sets across different donors, where we develop Cellular method? How can we integrate data sets across different species? Or how can we analyze data sets across different aging data? How can we analyze different aging data sets? But in this talk today, I would like to focus on how can we bridge data sets across different modalities. So I would present two methods that we recently developed that help us to bridge the transcriptome of a cell and the spatial organization of a cell, as well as a transcriptome of the cell and the molecular morphology of a cell. And I will also show some of the machine learning advances that we developed along the way. And in the second part of the talk, I would like to focus on the question, how well can we really predict the transcriptional outcomes of genetic perturbations? So, while single-cell RNA sequencing technologies enable us to study heterogeneity of cells at unprecedented resolution, the fundamental limitation is that cells are dissociated from the tissue and the spatial context is lost. On the other hand, we have spatial transcriptomics technologies that enable us to study cells in their native tissue context, but the number of genes that we can measure with spatial transcriptomics technologies is limited. So we ask the question, how can we overcome these limitations with the computational approaches? And can we somehow be able to get the best of both worlds? So ideally, we would like to have information about the spatial organization of the cells, as well as the full transcriptomic profile of a cell. So we started this project with a question, how well can we reconstruct complex tissue structures? So if we start from dissociated single cells, how well can we be able to predict tissue organization? And we call this process tissue reassembly because from dissociated cells, we would ideally like to be able to predict the organization of the cells, spatial organization of the cells. But the question is also to what extent is this tissue reassembly process even possible, given vast cellular heterogeneity, complexity of tissue structures, intricate cell-cell interactions, and also elusive biological mechanisms that underlie a tissue formation. And if it is indeed possible, at least in some biological systems, how can we learn these underlying rules of complex spatial organization? So the good news is that we have plenty of spatial transcriptomics data sets. So the idea here is that we could potentially leverage existing spatial transcriptomics data sets to learn a spatial prior using AI methods. So in order to do so, we need to be able to capture cell interactions locally, but also more globally. So with this goal in mind, we develop a method that we call Luna. And there are two key ideas in Luna. So the first key idea is that we model these cell interactions via attention mechanism. So attention mechanism is a powerful machine learning concept that allows model to dynamically focus on the most relevant parts of the input, and it is the building blocks of today's transformer architecture. So here the idea is basically that every cell learns how much importance should it give to other cells, that meaning kind of how much attention should it give to other cells in its context. So every cell learns how much importance to give to other cells based on the relevance of other cells to a cell's own location and gene expression. And this is a work developed by my PhD student Yeast and a master student Shanakia. So the second part of the Luna is that we model this tissue reassembly process as a generative process. So in particular, we use conditional diffusion models. What this means is that we condition the generation of cell coordinates on the gene expressions of the cells. And then with a number of diffusion steps, ideally, we would like to go from these noisy cell coordinates to the reconstructed tissue structures. So Luna leverages these existing spatial transcriptomics datasets, as I mentioned, with ground truth cell locations to learn proper priors. And as a diffusion model, it consists of two main steps. The first step is a corruption process where we add noise to the ground truth cell locations, meaning we sample diffusion time, and then from ground truth cell coordinates, we go to noisy cell coordinates. And the second step is a denoising process where we learn to recover the original cell coordinates through the denoising process. And to learn the cell embeddings, we adopt this multi-head self-attention mechanism that I introduced earlier. And then we go from these output cell embeddings, we predict the cell coordinates. We also design a loss that would be invariant to arbitrary rotations and translations of the input slice, but here I won't go into the details, but happy to discuss if someone is interested. Then during inference, what we do is we sample from standard normal distribution, and ideally with the number of diffusion steps, we would like to be able to reconstruct the tissue structures. So we first apply Luna to Murphy's mouse brain atlas dataset, which has over 4 million cells. So we train the model on 2.8 million cells from one mouse, and then we test it to 1.2 million cells from another mouse. So here on the left, I show ground truth locations. These are Luna's predictions, and this is how the fusion process happens in Luna. So I think there is a bit of delay because of the internet. But this is how actually model starts with these noisy cell coordinates, and then with a number of diffusion steps, eventually converges to meaningful tissue structures. Overall, we applied Luna to across entire mice brain, across 11 major regions, which are identified in the this Allen Brain Institute Atlas. And we generally see a good agreement between the ground truth and predictions. But we were next interested whether Luna, actually, given the high similarity between these two mouse brains, we were interested whether Luna would be able also to generalize if we exclude certain cell class during the training. So in particular, we excluded one of the 34 major cell classes one by one from animal one, and then trained the model on all classes except that class. And then during test data, we input all cell classes into the model, and we were interested whether a model would be correctly be able to position this unseen cell class. So here is an example from for glutamatergic neuron, where this class was excluded during the model training. And generally, we see that Luna was able to correctly position these cells within the spatial architecture of the tissue. If you look at the spatially variable genes, for example, TCHZ2 gene and SYT6 gene, we indeed noticed that the model was able to correctly predict which groups of cells, in which parts of the tissue these groups of cells that express these spatially variable genes should be located. So this was example previously for glutamatergic neuron by glutamatergic neuronal cells, but in general we see good performance for other cell classes. So this is another example for vascular cell class, which has highly different distributions. But again, the models, the ground truth locations and the model's predictions agreed well. So Luna is unique method in this idea to learn spatial priors across multiple slices and then generate locations conditioned on the gene expressions. Existing models either map dissociated cells to a reference dataset and rely on the exact spatial reference. So this would be example of the Tangra method. Or, for example, they have some assumptions. For example, in NovoSpark, the assumption is that cells that have similar gene expressions should also be spatially close together, close to each other. So the idea in Luna is that instead of manually kind of putting these assumptions, that we allow the model to learn these proper spatial assumptions. So we compare Luna to existing methods. And in general, this is a performance measure using Spearman's rank correlation based on the ground truth ordering of the cells. So we compare Luna across different slices from an unseen mouse brain. And in general, we see that Luna outperforms other methods. But even if we put prior information into other methods that Luna doesn't use about which spatial slice should is the most similar, what is the reference slice for the baseline method, so that the baseline methods have the most similar reference slice to which they can position cells. Luna still outperforms these existing methods, even though it doesn't use any such prior information. We next applied Luna for reassemble central nervous system single cell RNA sequencing atlas. We again use this model that was pre-trained on the Murphy's mouse brain, but now during inference, we input single cell RNA-seq data. Here we don't have the ground truth locations, but we estimate them using the data set where this single cell RNA-seq data was integrated with the StarMap Atlas. So we don't use StarMap Atlas during model training or anything. We just use it to estimate the ground truth locations. So in general, we see good agreement between the estimated ground truth locations. So this is an example of spatially variable gene, for example, IQGAP2, which shows a gradual downregulation across the Emmons horn region for hippocampus, which was well captured by the model. and MeFTC gene whose expression is actually minimal within the CA2 region, but is upregulated in isocortex and olfactory areas, which was also captured by the model. So why is this interesting? Because now we can potentially use Luna to identify unseen spatially variable genes. So this is an example where now Luna was able, because we can now embed the single cell RNA sequencing data, we can now ask the question, what are maybe spatially variable genes in the single cell RNA sequencing data which were not available in original spatial transcriptomics data sets? We next also look how can we possibly interpret these predictions of Luna. So one way to interpret the predictions on the gene level is that during training we can put all the genes, we can train the model on all the genes. And then during inference, we can remove gene by one, and then we can measure how much removing that gene affects the performance of Luna. Of course, here we cannot say anything about the causality, so but we can still try to interpret which genes may drive model predictions. So here we can basically measure what is the Luna's predictions when we include all genes, and then we can measure how much effect does removing a single gene, how much it affects Luna's predictions. So when we look at when we did this experiment across different cell classes, so here, for example, genes that pop up. So these genes are indeed genes that are closely tied to cell identity and function. For example, Claudin 11 gene encodes for oligodendrocyte a specific protein, which is and prior work has shown that mutant mouse that lack expression of this gene exhibit central auditory deficits and neurotransmitter imbalances. We next collaborated with Joele Lamano's lab at EPFL. Joele did experiments in his lab, and we used the Parkinson disease mouse model. In particular, alpha-synuclein was injected into the mouse brain, and these brains were collected 3 months post-injection. And then we had also control samples from a healthy mouse. And both of these experiments were done on the Xenium, using the Xenium technology. So we used the Murfish, this model that was pre-trained on the Murfish mouse brain atlas. And then during inference, we fine-tuned the model on the control samples of the Xenium data set. And then during inference, gave to the model this Parkinson disease mouse data to see whether a model could potentially be able to identify those spatial regions that could be affected by disease. So in particular, we removed completely the XY coordinates for the Parkinson disease data, mouse model data. Then we use Luna to embed control samples and the disease samples to the common coordinate framework. Because now, and Luna was used to infer locations of this disease data set. Then we identify those regions that have potential pathological alternations based on the differences in these regions on their gene expressions. And then in Joel's lab, they performed immunostaining for alpha-synuclein. And indeed, one of these regions that was identified by Luna corresponded to this region where alpha-synuclein protein was expressed. So the idea in Luna is that we try to bridge the gene expressions of the cells and the cell locations. But very recently, we also asked the question, can we link the cell morphology, in particular nuclear morphology of cells, with the gene expressions of the cells? And here, IRIS technology provides the chance. So IRIS is a technology that just, we put a manuscript on a bioarchive in December. So this technology has been developed by Barry de Planck lab at EPFL. So using IRIS, cells are loaded in the microfluidic chip. They are precisely placed, imaged, and encapsulated in a droplet to label cells mRNA molecules. So IRIS provides these paired samples of the cell morphology and the transcriptome of the cell. And in collaboration with Bart's lab, we were interested, can we now use this iris data to quantify the link and to establish relationship between the gene expressions of the cells and their morphology on a single cell level. So we developed, with this goal in mind, we developed Cosmic, that again was also the second part. So first, there is an Iris paper, and the Cosmic paper was actually put on arXiv in January this year. And the idea of Cosmic, which is a model developed by my PhD student Shuo and postdoc Ramon, is that we want to condition either we want to have the bimodal morphology to transcriptome model, where we were interested how well from the gene expression of a cell can we predict the morphology of a cell. In particular, nuclear morphology was the data that we had, but iris is generally applicable. We could also use it for cellular morphology. And how well, given the microscopy image of a cell, which genes would be predictable from nuclear morphology, and can we establish the link between the nuclear morphology of the cells and the gene expressions. So conditioned on the nuclear morphology of the cell, Cosmic generates the gene expressions of the cells, and then we try to identify those genes that could be predictable. And conditioned on the transcriptome of a cell, we generate the microscopy image of a cell. So in order to train Cosmic, we first needed the encoders of the nuclear morphology and the transcriptome of a cell. For transcriptome, there are plenty of existing models, for example, that one could use. So you could use, for example, SCVI, or you could do, if you want to do zero-shot embeddings, you could go with these foundation models like universal cell embeddings or single-cell GPT. But we also needed a strong nuclear morphology encoder. So for that, we trained our own model. So the good news is there's lots of nuclear morphology images that are publicly available. So we train our own large-scale model for nuclear morphology. So in particular, we trained this model on over 22 million nuclear microscopy images that we collected. They were collected from hundreds of thousands of different cell lines. Then we segmented these. We started with whole well microscopy images. Then we segmented to get individual nuclei. And then we trained this model in the mask autoencoder fashion. In particular, we would mask parts of the input patches and then train the model on these 22 million images to predict the patches that have been masked. And then we use this pre-trained model to learn to get the embedding of the nuclear morphology. And using this embedding, we then condition the model to generate the gene expressions of the cells, and vice versa, condition on the gene expression of the cells, we train a diffusion model to generate the microscopy image of a cell. And in order to train this bidirectional relationship, we leverage the iris pair data. So next I will show some examples of using Cosmic to go from the transcriptome to the nuclear morphology of a cell. So we've evaluated the model by comparing generated and real images in this space of the morphology, large-scale morphology model that we train. So in particular, we take the ground truth images, we get the image embedding, we take the generated images generated by Cosmic, and then embed both of these images into the embedding space of this large-scale nuclear microscopy image model that we pre-train. So now this is the embedding space of the ground truth images. This is, of course, two-dimensional UMAP projection, starting from this large high-dimensional image embedding. And this is the Cosmic generated space. So if we sample some of the cells from this space, we see in general visually the cells, the model is capturing the differences between different cell types well. This is not the case, for example, for other models that could be applicable for this task, like multi-domain translation and MorphNet, where both of these models actually don't resemble so much the ground truth image space. And also if you measure quantify the differences in generation between Cosmic and these methods, we quantitatively see a large difference. So this is just an example how ground truth images look like across across different cell lines, and how cosmic and MorphNet and multi-domain translation images look like. Next, we were interested to further quantify this. So the idea here is that we could potentially train the model on the ground truth images, and then during inference, so we never train the model, or model has never seen these generated images. The model has always been trained on the ground truth images, but now during inference, we can input, for example, images generated using different models to see how well the model could distinguish between different cell types. And here we see the differences. We see actually that images that were generated by Cosmic, the classifier trained on ground truth images, was able to almost get the performance as good as the performance you would expect on the ground truth images, which is actually the upper bound, while there is actually a big gap between other methods in this scenario. We were next interested how well can the morphology of a cell, how well the transcriptome of a cell could be predictable from the morphology of a cell. So we again use the iris pair data, but now we use the other direction of the cosmic. Now this is the gene expression space. So the cells in yellow are the cosmic generated cells, and these blue, the cells in blue color are ground truth cells. And this is now the space colored based on different cell types. If we compare again Cosmic to other methods, it's much better, but interestingly, there were about more than 15% of genes that have correlation coefficient higher than 0.4. But so far we used mainly cell type information to evaluate the results. So we were interested also to look further. Does Cosmic learn also information beyond the cell type information? So what we did is that for each cell type, we randomly reassign transcriptomic profile to microscopy images, preserving the exact sets of images, expression profiles, and labels while breaking the true pairings. Thereby, we removed any linkage between morphology and the gene expression within a particular cell type. So if the model relied only on the cell type information to make predictions, then gene level performances would be similar between original and permuted data, which we indeed observe for the marker genes. So marker genes were predictable before and after permutation, which is expectable. But interestingly, there is a group of cells actually that is highly affected by this permutation. So they were highly predictive before the permutation, but not anymore after the permutation. And when we look closer, for example, these genes included cytoskeletal and contractile regulators. And also there were some as well, other genes were also cell cycle genes. We were then also further investigated how well could model distinguish between different cell cycle phases. But in the interest of time, I won't go deeper into that, but I would like to show actually how Cosmic could also be used potentially to find a nuclear morphology related gene. So in particular, what we could do is we can start with the ground truth transcriptome, then we can use Cosmic sequence to image to generate the nuclear morphology, and then Cosmic image to sequence to back generate the transcriptome of a cell. Then we can calculate the percent correlation for each gene, and the idea is that only nuclear morphology related genes can pass this information flow. However, again, this is not causal. We cannot say anything about causality. We don't have perturbational data. It's more here establish the correlations between the two. So in collaboration with Walter Karphaus lab at EPFL, we applied this to DO145 prostate cancer cell line. So in particular, we treated DO145 cells with the chemotherapeutic agent docetaxel and profiled then non-treated cells and the docetaxel treated cells. We observed that actually among those treated cells, there were subpopulation of cells that grouped together with the non-treated cells, while another subpopulation cells was forming its own cluster. So this is all in the space of this large-scale morphology model that we pre-train. And then we were, indeed, when we look at the nuclear morphology of these cells, we saw that those docetaxel responsive cells had in large nuclei with reduced solidity. where resistant cells likely continued, and the assumption is basically that the resistant cells likely continued cycling normally, and as a result retained a more retained more regular nuclei, while docetaxel treated cells, those that were forming its own cluster and have changes in the nuclear morphology, they were probably in G2M arrest. So we next applied Cosmic to this idea of trying to identify those genes that could be associated with nuclear morphology of a cell as a proof of concept. And applying this framework to docetaxel treated cells and untreated cells, we identify a set of genes which are for this set of genes include CKS2, where was the gene that had the highest score, and the second one was BUB1 gene, and all of these top 5 genes are actually genes that are well-known drivers of the tumor progression. For CKS2 gene, for example, we also noticed that there are 2 clear gene expression subpopulations that are also correctly modeled by Cosmic, and they correspond to these potentially responsive and non-responsive cells. So while working on a Cosmic, actually the fundamental problem that we had is that the number of the multimodal samples was not as huge as typically is when you build the multimodal foundation models. So in order to kind of have a general framework that would allow us to work with these small amount of the multimodal samples, we were wondering, can we propose like a general framework that could be applicable for even beyond biology. So when we think about these days, about how we build these multimodal foundation models, in general in machine learning, these models are trained on tens or even hundreds of millions of paired samples. However, these millions of paired samples are often unavailable in many biomedical domains. So we asked the question, can we create these powerful multimodal models even if we have limited amount of the pair data? So imagine you have like samples from modality 1 and modality 2, and now your goal is to build the alignment between these 2 different spaces. But the question is, how can we guide the model toward a well-aligned solution? So the typical approach often is that we could train from scratch using plenty of multimodal data. But as I said, in biology, very often we don't have these huge, tremendous amounts of the multimodal data. So alternatively, what one can do is what we did in Cosmic, is that you can try to start with some pre-trained unimodal models. And now the question is, how can we effectively align these unimodal models, even if we have limited amount of the multimodal data? So existing approaches in this space, and now this is kind of very generally in the AI space, is that if you had, for example, models from modality 1 and modality 2, for example, vision model and the language model, now you would froze these encoders, and then you would train the alignment function. For example, the alignment function could be contrastive learning objective, which forces samples that are similar to each other to be close together, while samples that are different to be far away from each other. But even if you adopt this contrastive learning objective in existing machine learning literature, we still require millions of paired samples. So we asked the question, can we use less than 1% of data typically used in the field to still build multimodal models? And this work has been done by my visiting PhD student Fabian, my PhD student Shuo, and the master student Huyen. So the key idea for us is that in order to build a strong alignment, the idea is that we want to preserve the structure of the pre-trained unimodal space. So in particular, what we introduce is that in this unimodal space, we look at the neighborhood of samples in this space. And now during training the multimodal models, we actually want this neighborhood to still be preserved. So in particular, we introduced this very simple structure regularization, where the idea that we want to preserve the structure of the pre-trained space, so the data points that are similar to each other in the pre-trained space should remain similar after multimodal training in the aligned space. So in particular, we can start with these pre-trained spaces. We can form similarity matrices, then transform them to similarity distribution. And what we do is compute the Jensen-Shannon divergence in order to and we minimize Jensen-Shannon divergence in order to preserve the structure. So this structural regularization basically captures these kind of relationships, neighborhood relationships. And the idea is that this regularization can actually be simply added to existing contrastive learning objectives. So we add this regularization for modality 1 and for modality 2. And it's, as I said, like it can also be no matter what alignment function you have, you can just add this regularization that preserves this neighborhood structure between the unimodal and the multimodal space. So interestingly, we show that using this structure regularization, we achieved average 68% improvement for linear alignment across general computer vision datasets for zero-shot classification as well as retrieval. And for MLP alignment, if you want to do alignment using nonlinear alignment, the improvement is 75% across different datasets. And again, we trained these multimodal models using only 80,000 samples, which is actually less than 1% of data typically used in the field. And most recently, this is also just briefly, like very recent work that we are actually very excited about. There was lots of discussion in the AI community about this Platonic representation hypothesis, which claims that neural networks, if you train them with different objectives on different data and modalities, are converging to a shared statistical model of reality in their representation space. So this was proposed by Philip Isola at ICML 2024. And there was lots of excitement in the community. So what this means is basically that if you have a, let's say, language model, you have, and you measure the similarity of the language model with vision model representations, what we see is actually that larger the model size, the better the agreement between the embeddings of a vision and the language model. So we recently revisited this Platonic representation hypothesis, and actually we show that these existing representational similarity metrics that have been used in the field to measure the similarity of the embeddings are actually confounded by network depth and network width. So this means that the deeper the network, you expect to see higher similarity, and also the wider the network, you expect to see higher similarity. So we proposed a calibration framework that actually adjusts for this. And then we show that after calibration, there is no more global alignment. So basically, metrics like, for example, center kernel alignment (CKA). There is no more. It's no longer the case that the bigger the model, the better the alignment. Actually, we don't see any correlations anymore. However, the local similarity still holds. So metrics such as mutual KNN, for example, which measure the local similarity of the embeddings, actually it still holds that the bigger the model, the better the alignment. So based on these findings, we revisit this original Platonic representation hypothesis, and we propose our alternative Aristotelian representation hypothesis, which says that neural networks, if you train them with different objectives on different data and modalities, what we can only claim is that they are converging to the shared local neighborhood relationships. And this work was actually put on arXiv very recently. And finally, in the last part of my talk, I would like to also discuss shortly our recent work on where we try to answer the question, to what extent can we predict the transcriptional outcomes of unseen genetic perturbations? And this work has been led by my postdoc Ramon. So understanding how genetic perturbations affect single cells is crucial for advancing functional genomics with a wide range of implications, including revealing gene function, mapping regulatory networks, cell engineering, and so on. So the question in particular is given groups of control cells and a perturbation, what would happen if we were to perturb cell in a single cell in a certain way? Ideally, the goal is to predict the outcome of genetic or chemical perturbations on the transcriptome of single cells. So here I will particularly focus on genetic perturbations. And recently there have been works that have been claiming that they can predict the perturbational response to unseen genetic perturbations. What does this mean is that you train the model on a set of perturbations, and now during testing the model, there is a new gene that has not been seen in your training data set, and you try to predict the transcriptional outcome of perturbing this gene. How is this possible? Of course, we need some prior knowledge. For example, existing foundation models use as prior knowledge this idea of training across tens of millions of single cells. And this prior knowledge could potentially then be leveraged to predict the predictions, to be able to generalize to unseen perturbations, or more specialized models that use, for example, prior information about a functional similarity of genes. So we studied actually to what extent is this really the case, and to what extent can we predict transcriptional outcomes of unseen genetic perturbations. So we collected about 10 different data sets across 3 different technologies and 5 different cell lines. And we adopted standard metrics that have been used in the literature, including Pearson correlation between predicted and ground truth expression changes of a perturbation with respect to the control cells. So we can measure this correlation on all genes, or for example, on top 20 differentially expressed genes, as it has been done in the literature as well. So we constructed this very simple baseline that we call perturbed mean, which is just taking the average expression of all perturbed cells as the prediction. And then this profile is used as a prediction for all unseen genetic perturbation. So surprisingly, we find that this very simple baseline called perturbed mean, and again, it makes same predictions for all unseen genetic perturbations. It is actually outperforming or achieving very similar performance to these much more advanced state-of-the-art methods. So for we were then wondering whether it's also the case for combinatorial perturbations. For combinatorial perturbations, we can also incorporate some priors. So we constructed also a very, very simple baseline. So if I haven't seen any of these 2 genes being perturbed, I just take as my prediction this average as before. If I have seen one of these genes being perturbed in my train set, then as a prediction, I will take this average of this perturbed mean and the average of expression of cells that underwent the same type of perturbation in the trained set. So again, I'm just taking averages and taking averages from my trained data. Or matching mean, if I have seen both genes being perturbed, I can just take the average of all those cells that underwent the same type of perturbation. Now what we see for the Norman data set, with combinatorial perturbations, which is one of the most benchmark data set in the field, we again see that actually this very simple baseline outperforms state-of-the-art methods across 0, 1, and 2 seen perturbations. So we were very puzzled by this. So we were questioning why this happens. Why do simple baselines perform so well? So we hypothesize that this comes from systematic differences between control and perturbed cells that could be caused, for example, by potential selection biases or, for example, underlying biological factors. So we indeed investigate this across all different perturbation response prediction data sets. I will just show an example for replogal data set. Because this is a large scale perturbation screen data set. And we were actually surprised that there are also systematic biases even in this data set. So for example, in this case, it is underlying biological factors that are potentially causing this systematic variation between the control and perturbed cells. So we observed a significant difference in the distributions of cells across different cell cycle phases between perturbed and control cells. And this can be attributed to the fact that widespread chromosomal instability is induced by perturbations of that these RPE1 cells with abnormal karyotypes tend to react to the chromosomal instabilities that are caused by perturbations through cell cycle arrest, which causes different distributions of cells across different cell cycle phases. So next, we wanted to quantify this systematically, this degree of systematic variation. So we define perturbation-specific shift as a vector that points from the centroid of control cells to the centroid to this perturbed mean. And then we define these perturbation-specific shifts as vectors that point to from the centroid of control cells to the centroid of cells that underwent the same type of perturbations. Now we can measure the cosine similarity between these vectors, and this is how we define the systematic variation. And the idea here is basically that high cosine similarity indicates that the transcriptional responses to different perturbations are aligned in a similar direction, which suggests shared, possibly non-specific shifts in the gene expression. So when we quantify the systematic variation in such a way, we indeed see that, for example, very frequently benchmark data set Adamson and Norman have a high degree of systematic variation. Which is not actually taken into account by existing perturbation response prediction methods, because commonly used metrics are actually influenced by this systematic variation. So in particular, what we observe is that if you measure this changes using Pearson correlation coefficient, the higher the Pearson correlation coefficients is actually the higher the value of the score corresponds also with the degree of the systematic variation. So there is a high correlation with the degree of systematic variation in a particular data set and the eventual performance of this on the data set using this matrix. So this shows and indicates that these metrics that we have been using so far can lead to inflated performance under data sets that have systematic differences. So in order to adjust for this, we propose a Systema evaluation framework, which the idea is that we change the reference instead of using the centroid of control cells as a reference, we used the perturbed mean as a reference. So now changing the reference to the centroid of all perturbed cells can emphasize these actually perturbation-specific effects, and we can look at the shifts from perturbed reference in order to capture these perturbation-specific effects. Now, if we change the reference and look at the Pearson correlation coefficient, we actually see that the scores are pretty low, and that predicting perturbation-specific effects for unseen genetic perturbation is actually a remarkably challenging task once we adjust for systematic variation. Now, also with this adjustment, we see low correlation with the degree of systematic variation across all datasets, which is also what we would expect. We also introduce a simple metric called centroid accuracy, which tells us for a given perturbation, is the predicted centroid closer to the actual centroid or closer to the other centroid. So this is kind of intuitive metric that measures whether predicted post-perturbation profiles are closer to their ground truth centroid versus the centroid of other perturbations. And the centroid accuracy of 1 indicates that the inferred profiles recover the expected transcriptional effects of a perturbation. We measure the centroid accuracy now across different data sets. These black lines show the performance of this perturbed mean. And we see that on some data sets, actually the fine-tuned version of the single-cell GPT does outperform this baseline, but it's not the case across all the data sets. So now this actually metric is also useful because it can allow us to divide the predictions in those that are informative and uninformative. So you can, for example, take method of your choice and measure the centroid accuracy of a method of your choice. And then on the y-axis and then on the x-axis, you can measure the centroid accuracy of perturbed mean, this very simple baseline. And then you can ask which predictions is the my method or the method I'm testing, evaluating better than this perturbed mean. When we do this, for example, for a single cell GPT, we see that those genes for which the method actually performs better than this perturbed mean are ribosomal protein genes, which are highly conserved and co-regulated gene families. So finally, this work has been done really by my amazing team, my amazing PhD students, postdocs, and also amazing collaborators. So with Bart, we collaborated on the Iris and Cosmic papers, with Joelle on Luna, and with Sarah and more on Systema. Thank you so much for your attention, and I'm happy to take questions. And also, not to forget, we are also hiring. So if you're interested in doing a postdoc in the lab or a PhD, being a PhD student, please reach out.
Thank you very much indeed, Maria. So I think we have a plan for how to get you questions. Either unmute yourselves or type them in the chat, please. I've got one that's been here for a little while, so maybe I can start with that one. Steph Davis asked, how can this tool be used for cancer stem cells in the brain?
Uh, sorry. Yes.
So how
Mhm.
can your sort of machinery be used for cancer stem cells in the brain?
Machinery Luna or
Yeah, so let me see. Is Steph still here? See if she's
I'm not sure which machinery.
yeah, I'm just trying to see if she's can't tell if she's still here or
Maybe she can also explain what exactly would she like to do. Nice to see you, Bianca.
no, I think she's not here. So we'll move on. Who's next? Okay, here's another one. So in predicting spatial features of unseen cell types, which types of cells are difficult to predict, and what are the features of these difficult cells?
Um, so in general, I would say so. For example, for these unseen cell classes, we excluded like one of the 30 major cell classes. And across all these cell classes, we generally see good performance. What we see, it's more difficult if the number of cells is also kind of like much smaller. I'm not sure actually we don't have actually we didn't see like observe like significant differences in performance across different cell classes. Um, like that we could say that one is like really unpredictable versus other was like much more highly predictive. But about exact rankings, I'm not sure about exact rankings, but we didn't see like huge differences in the predictive performance of the model.
Any more questions? Oh, any more questions, anyone? You can unmute if you want to ask the question directly. So Lorenzo, can you see any others?
No, not right now.
All right. Then I think that's maybe a good time to stop. Thank you very much indeed, Maria. That was wonderful. And
Thank you.
as has been noted before, you have to come in real time next time. So
Yes,
congratulations, and we'll hope to see you again.
thank you. Hope it was useful and hope people enjoyed it.
That's fantastic. Thank you.
Thank you.